Today we'll be talking about the Cre-Lox system and how you can create knockout models of animals using the Cre-Lox system. The benefit of the Cre-Lox system over the traditional knockout models is that the Cre-Lox system can knock out genes in only specific types of cells that either come from certain cell lineage or are under the control of a promoter that can be inducible, or they're active in certain cells that can only at a particular developmental stage. And now we'll talk about the mechanism of Cre-Lox system.
So in order to get a mouse, transgenic mouse with the Cre-Lox system activated, what you have to do is you get a transgenic mouse with the Cre protein and controlled by a promoter that's either tissue dependent, inducible, or time dependent at some point in development. And then you also have your transgenic mouse here that has your target gene surrounded by two LOXP sites. You may notice that these sites are directional. They're both facing one way, and that's very important for the excision of DNA. If they were both facing opposite directions, then you'd get inversion of the DNA instead of an excision. So that's very important there. So when you have your two transgenic mice, you cross them, and then you get a progeny mouse down here that has not only the Cre protein regulated by a specific promoter, but also your target gene with the two LOXP sites. As I said before, the Cre protein is regulated by a promoter that is either inducible or tissue dependent or time dependent. So it can only be expressed in certain cells, certain times, or when you want. So what you end up getting is a mouse that can live through development, and then you can induce this Cre protein to get a deletion mutant for your target gene. So how that exactly works is, if you look at this model over here, you see this is your target gene, and then you have your two LOXP sites. Notice that the arrows indicate the directionality of the LOXP sites. And then you get your your recombinase dimerizing on the LOXP sites. Imagine that these proteins are actually much smaller for your viewing pleasure, but in reality this is much closer together. So when you get these dimerizing on the LOXP sites, what happens is there is catalytic activity that actually cuts the DNA here and here, and you get this association with this tyrosine to create phosphotyrosine intermediates. What this does is this actually brings the phosphate close to the cut DNA, so you can get this 5' 3' binding of the DNA on both sides. And now what you have is a very allosterically inhibited intermediate molecule. So what happens here is, again, you get this catalytic activity to cut the DNA, and you get the formation of the phosphotyrosine intermediate molecule. Here and here. And then again, this is now very close spatially to this cut DNA, which will then associate here, 5' 3', and here. And what you have now is your excised gene with the LOXP site attached to it. And up here you have the rest of your DNA separated from it.